This presentation about slime molds was created by Amy Kudela, Catherine Myers, Kristen Schreiner, and Kirsten Greenfield, and it was created for the Biology 1520 class at Georgia Tech. Slime molds are fungi-like heterotrophs that are important in the decomposition of organic matter in the temperate and tropical forests. For the longest time, slime molds were thought to be a type of fungus because they act much like them in that they decompose dead material for food and reproduce through spores. The main differences are that slime molds belong to the kingdom Protista, they do not have hardened cell walls, and they move. The two primary types of slime molds are plasmodial slime molds and cellular slime molds. As discussed in lecture earlier in the semester, there are organisms that only produce one sex of offspring unless the environmental conditions are harsh. This is similar to how the environment affects slime molds' life cycles. The life cycle of both plasmodial and cellular slime molds can be seen as divided into two parts, a feeding and growth phase that is motile and a reproductive phase that is stationary. During environmentally ideal situations, cellular slime molds are single motile, amoeba-like cells that function independently from one another. When environmental conditions become dangerous for the slime molds, they begin to self-organize into a single body called a pseudoplasmodium, or slug, that will turn into a fruiting body that produces spores. The spores contained in the capsules are released and hopefully well dispersed into wet places. They are then allowed to germinate, releasing single amoeba that begin their independent life cycle. During this, two of the haploid amoebas can reproduce sexually by fusing and producing more haploid individuals. This can continue until the environmental conditions require aggregation again. During this aggregation process, the cells follow the chemical gradient of cyclic adenosine monophosphate, or CAMP, found in their environment, accumulating in the CAMP-dense area. Once the environmental CAMP concentration hits a certain threshold, the slime mold cells start to release the chemical signals themselves. CAMP signals lead to the aggregation of slime molds into one multicellular body. CAMP, along with other chemical signals, help differentiate the different types of cells in the aggregate body. The body differentiates into pre-stalk cells that move to the anterior end of the body and pre-spore cells that move to the posterior end of the body. The pseudoplasmodium has a specialized head that is heat and light sensitive so that the organism can move quickly to a suitable environment. Once this environment is found, the pseudoplasmodium stops moving and culminates. The lack of a food source and the presence of light allows the cells at the anterior and posterior end to differentiate into the specialized cells that become the stalk and the spores of the fruiting body. The mobile phase of a plasmodial slime mold is called a plasmodium, which is a single multinucleated cell that in some species can be a meter in diameter. The plasmodium is able to move towards food sources and away from light to grow in favorable conditions. When unfavorable environmental conditions are reached, the plasmodium travels to a dry, well-lit area to begin reproduction. This is a very regulated process that includes the complete conversion into a stationary organism known as a fruiting body. Haploid spores are produced inside the fruiting bodies by meiosis. The germinated spore can transform into either an amoeba-like cell or a flagellated swarm cell. The amoeba-like cell can reversibly turn into a flagellated swarm cell in liquid environments or create cysts upon desiccation. Cysts can transform into flagellates if the cell wall isn't fully developed before the re-exposure to liquids from a dry state. Two haploid amoeba-like cells of different mating types may join together in a cellular fusion followed by a form of nuclear fusion. This is a very primitive, basic form of sexual reproduction, but creates genetic diversity nonetheless. The fusion of two haploid cells results in a diploid zygote, which transforms into a developing plasmodium by nuclear division in the absence of cellular division. The absence of cellular division creates the multinucleated motile single-celled Plasmodium. A study was done to uncover the patterns of movement of slime molds. A maze was created with a slime mold at one end and a food source at the other end. The study showed that at first, the mold spread somewhat randomly towards the food source, taking many pathways. The paths that were taken and were unsuccessful in locating the food were glazed in a thick mat of non-living translucent slime. This slime allowed the mold to know where it had already searched. As the slime network grew, it narrowed the maze down to one pathway that was successful in the food uptake. This concept was later applied in Tokyo for a real-world problem. When the Tokyo Railroad system was built, the constructors used slime molds to create the most efficient system. They put a food source on a map at the location of the cities needed to connect the railway system and introduced the slime mold. The slime mold spread throughout the map in multiple configurations, much like while in the maze. After unsuccessful pathways were removed from the map, the shortest route between the food sources was highlighted by the slime mold and used as the path for the railway system. The structure of slime molds allowed them to be used in many applications. Because plasmodial cells tend to be very large, they are easily manipulated and they are used in studies that view the movement of intracellular contents. Cellular slime molds are also easily manipulated, and their unique aggregation process is used to study cell-to-cell interaction when single cells generate multicellular organisms.